Good morning, children. I hope you all are fine. Yesterday you have responded well. All of you have attempted the Google Form MCQ and you did well also. In the previous class, we have studied about dihybrid cross. What is a dihybrid cross? It is a cross between two plants that differ in two pairs of character or two pairs of allele. So which plant was selected? Pisum sativum. In this two characters selected were, one is the shape of the seed, that is round, color of the seed, that is yellow. These two are dominant. So capital R, capital R, capital Y, capital Y. And the other parent selected is having wrinkled seed with a green color. So how to represent recessive character? Small r, small r, small y, small y. So gametes are haploid. So one gamete is having capital R, capital Y. Other gamete has small r, small y. When these two gametes fuse with each other, we got F1 generation, that is round and yellow. Phenotypic condition heterozygous round yellow, that is capital R, small r, capital Y, small y. When two F1 generation plants were self-pollinated or self-crossed, in the F2 generation we got the ratio of 9:3:3:1. That is 9 plants were having yellow and round seed, 3 were having yellow wrinkled seed, 3 with green round seed and only 1 with green and wrinkled seed. So in the F2 generation, in a dihybrid cross, phenotypic ratio is 9:3:3:1. In a monohybrid cross, phenotypic ratio is 3:1. So the unique feature of a dihybrid cross is here, along with the parental combination, we got new combinations or they are called as recombinant. Parental combination is of round yellow, wrinkled green. But here we got round green as well as wrinkled yellow. In which generation? In the F2 generation. So along with the parental combination, we have got new component, that is called a recombinant. After dihybrid cross, Mendel has formulated the third law of inheritance, that is called law of independent assortment. Can you recollect? First law is law of dominance. Second one is law of segregation, that is the universally accepted law. Now after dihybrid cross, Mendel has formulated the third law of inheritance called as law of independent assortment. This law states that when two pairs of traits are combined in a hybrid, segregation of one pair of character is independent of the other one. That is, when we are combining two pairs of trait. What are the two pairs we have combined? Round yellow and wrinkled green. Okay, when this law states that when two pairs of traits combined in a hybrid, so we have combined, we have crossed round yellow with wrinkled green, and in the F1 generation we got round and yellow. Now in the F2 generation, separation of one pair of character is independent of the other one. The meaning is, we have taken round yellow, wrinkled green. If these two are not moving independently, if these two are not separating, then in the F2 generation also we have to get which two character? Round yellow and wrinkled green. But in the F2 generation, we got round yellow, we have got round green, we got wrinkled yellow as well as wrinkled green. That is what the law says. The law states that when two pairs of characters are combined in a hybrid, separation or segregation of one pair of character is independent of the other. That is why along with the parental combination, we are getting new combination. What was the new combination we got? One is of round green, another one is wrinkled yellow. That is this law is called as law of independent assortment. It states that when two pairs of traits are combined in a hybrid, segregation of one pair of character is independent of the other pair of character. This is the third law of inheritance. Now let us pass on to a dihybrid test cross. You know what is a test cross? Test cross is crossing an unknown genotype with the recessive parent. Why with the recessive parent? Because for the recessive parent, the recessive allele always express itself under homozygous condition. So we know the genotype of the recessive plant, right? That is why we are not crossing with the dominant one. We are always crossing with the recessive one. In a dihybrid test cross, the ratio is of 1:1:1:1:1. So here you can see this is small r small y. So we will be getting one round yellow, another one is round green, wrinkled yellow and wrinkled green. So this is called a dihybrid test cross and the ratio is 1:1:1:1. Now why Mendel's work remained unrecognized till 1900? Because first reason is lack of communication. So now whatever happened in one part of the world, very next minute we can know, because communication is very fast. But during his time there is lack of communication. Second one, he used the word factor. Later on it is called as gene. That was not accepted by his contemporaries. And the third reason, already you have studied that he has used the mathematical logic. So use of mathematics in the study of biology was not accepted during his time. And during his time, he could not give any physical evidence for the existence of gene or factor. Because of these reasons, his works remain unrecognized till 1900. One is lack of communication. Another one, the concept of factor or gene is not accepted by his contemporaries. Use of mathematics in the study of biology was not accepted during his time and he could not provide any physical evidence for the existence of gene or factor. Now the next part is about the chromosomal theory of inheritance. This theory was proposed by two scientists, Sutton and Pavry. They have the one first introduced chromosomal theory of inheritance. So they have studied the resemblance between the chromosome and the genes. So both chromosome as well as the gene always occur in pair. For example, if I asked you how many chromosomes do you have, you will tell I have 23 pairs of chromosome. That means both chromosome as well as the genes occur in pair, because you know very well that each gene consists of two alleles. Two alleles of a gene pair are located on the homologous site of homologous chromosome. Last year you have studied what are homologous chromosome. So the two alleles of a gene pair are always located on the homologous site of the homologous chromosome. The pairing as well as the separation of the chromosome lead to the separation of the alleles of a gene also. So this is called as which one? Chromosomal theory of inheritance. That is, the behavior of the chromosome and the allele pair of a gene are parallel to each other. They both always occur in a pair. The allele pair are located at the homologous site of the homologous chromosome. During cell division, separation of chromosome lead to the segregation of the allele pair. This is what is called as chromosomal theory of inheritance. It was proposed by Sutton and Pavry. So here you can see that chromosomes are moving with each other. So next scientist is of Thomas Hunt Morgan. He studied the inheritance pattern in case of fruit fly. The scientific name of fruit fly is of Drosophila melanogaster. T.H. Morgan. The experimental material used by him is of to study the chromosomal theory of inheritance. He conducted several experiments in an organism called as Drosophila melanogaster. Common name is of fruit fly. So why he has taken only this particular fruit fly or Drosophila? Because it can be grown in any type of synthetic medium, easily grown in synthetic medium. Second thing, it completes its life cycle in about 2 weeks time. Within 2 weeks you can get the result. In a single mating, it can produce hundreds of progenies, large number of progenies it can produce, and there is a clear cut differentiation of sex. Male and female fruit flies are easily differentiated, they are distinguishable, and it has many pairs of contrasting characters. Clear why he selected this particular fruit fly? First reason is that it can be grown under any type of synthetic medium, complete its life cycle within 2 weeks time. So we can get the results within 2 weeks time, and it has a very a single mating, it produces hundreds of progenies, sexes are separated, and they have many pairs of contrasting traits. That is why he used this particular insect for his study. So next class you will be studying about linkage and crossing over. So today you have studied what is a dihybrid cross. Cross between two plants that differ in two pairs of characters or two pairs of alleles. The characters selected are round yellow, wrinkled green, F1 generation, heterozygous, round yellow, F2 generation. We got the ratio of 9:3:3:1. After this, Mendel formulated his third law of inheritance, that is called the law of independent assortment. It states that when two pairs of characters are combined in a hybrid, one pair of character segregate or move independent of the other one. That is the reason why in a dihybrid cross, along with the parental combination, we get a new combinant or recombinant also. Next you have studied about the dihybrid test cross. Crossing the unknown genotype with the recessive parent is called a test cross. The dihybrid test cross ratio is of 1:1:1:1. Then next you have studied why Mendel's works remain unrecognized till 1900. One is because of lack of communication. Second one is the concept of gene or factor was not accepted by his contemporaries. Use of mathematics in the study of biology was not accepted during his time. And fourth one, he could not provide any physical evidence for the presence of gene or factor. Then next you have studied about the chromosomal theory of inheritance. It was proposed by Sutton and Pavry. They considered that the behavior of chromosome as well as the gene are parallel to each other. They both always occur. The allele pairs of a gene are always located in the homologous site of homologous chromosome. The pairing and separation of chromosome lead to the separation of pair of gene or factor they carried. For the chromosomal theory of inheritance, experimental proof was given by a scientist T.H. Morgan. The experimental material used by him is fruit fly. Scientific name is Drosophila melanogaster. Why he used this particular organism? Because one is it can be grown in any type of synthetic medium. Second one, complete its life cycle in about 2 weeks time, so we can get the results quickly. In a single mating, it can produce more number of progenies. Sexes are separated and they have many pairs of contrasting traits. So thank you children. Go through up to this. Let us meet tomorrow. Thank you.